Click the bell icon and subscribe to our channel so that you will not miss any classes. Hello, welcome to class. Now we shall continue with the phases of meiosis 1, that is the metaphase 1. Metaphase 1. We have completed with the phases of prophase 1, starting from leptotene up to the diakinesis. I will draw a cell which is in diakinesis so that it will be easier to understand what happens in the metaphase 1. In diakinesis, what happened? There is a complete disappearance of nuclear membrane and the crossed over chromosomes were separated like this. Crossed over chromosomes were separated and there is a complete disappearance of nuclear membrane. As a result, the chromosomes are suspended in the cell cytoplasm. In the cell cytoplasm, the chromosomes were suspended. This is the condition in diakinesis, after completion of diakinesis. When the cell enters into metaphase 1, so here also there is a complete formation of the spindle fibers. Spindle fibers are being formed like this, and nucleolus is disappeared completely. The nuclear membrane is disintegrated into vesicles, and there is a disappearance of all other cell organelles. And by the end of this, the cell will enter into the next phase of division, that is the metaphase 1. The characteristic feature of metaphase 1 is there is a formation of cell plate, or we will call it as metaphase plate, and there is a formation of metaphase plate, and there is a completion of spindle formation or completion of spindle assembly. Spindle assembly is complete, and during the metaphase plate formation, the chromosomes forms two parallel plates on N chromosome on one side, which means normally we have studied from the meiosis that all the chromosomes will align in a center plate like this. I am discussing of mitosis. So all the chromosomes will align in a metaphase plate. Whatever the number of chromosomes present, they align in a single line. But in meiosis metaphase 1, the condition is quite different, where the homologous chromosomes, the n number of chromosomes will form one plate, and the other n, because the cell is diploid now, so n number of chromosomes will form one plate and other n will form another plate, so that there is formation of two parallel metaphase plates. So I will write like this. So this is one. Here is the crossover region, and the other chromosome will be here. So here is the one plate, one metaphase plate. The homologous chromosomes of these two will be lying on other side. Here they will be lying like this. So how many plates are formed here now? There is formation of two parallel metaphase plates like this. So this is the fascinating thing which is happening in the metaphase 1 of meiosis. There is formation of two parallel metaphase, and this plate will represent n number of chromosome, and this will represent n number of chromosome, totally 2n. This cell is still 2n in condition. And of course, there is presence of two kinetochores for each of the chromosome. We studied the structure of chromosome previously, and we know that on the either side of centromere, there is presence of two kinetochores which facilitate the attachment of spindle fibers. And here there is a formation of spindle operators already, and there is an attachment of spindle fibers to the chromosomes. And here the attachment is very different. How it is different is the spindle from one pole will attach us to the kinetochores of the chromosome. That is, I'll write it here, two kinetochores of each chromosome, attaches to the same pole spindle, which means there are two kinetochores here and both of them are attached to the same pole. The condition is different in mitosis. Mitosis, what happened? Here is the spindle from one pole will attach us to the one kinetochore. The other kinetochore will be attaching to the opposite pole. This is the condition in mitosis. But here in meiosis 1, that is in the metaphase 1 of meiosis, the chromosomal kinetochores, both of the kinetochores of each chromosome are attached to the same pole spindle. So that is the condition. All of them will be having the same thing. So here is the attachment and here is the attachment, like this. So this is how the metaphase plate is formed here. So the metaphase plate formation is the peculiar thing in the metaphase, as we know. But here there is a quite different metaphase plate formation. In case of meiosis 1, metaphase 1, there is formation of two parallel metaplates where each of them are representing the n number of chromosomes, and each of the chromosomes are attached to the same pole spindle. That is, the two kinetochores of same chromosome are attached to the same pole spindle, not of the opposite. So that is, these are the things which are happening in the metaphase 1. And after metaphase 1 is anaphase 1. Anaphase 1. And here in anaphase, since the chromosomes are attached to the same pole, there is a movement of chromosomes towards the opposite pole. Here is the movement. anaphase, what happens? The complete chromosomes are moved to the opposite pole due to the contraction of spindle fibers. What happened in mitosis? In mitosis, the chromatids are split at centromere, and from there they are dragged towards the opposite pole. That is, the chromosomes are getting separated and they were moved to the opposite pole. But here, the chromosomes are not separating. Whole chromosomes are moved to the opposite poles. That is, n number of chromosomes are moved to the opposite poles. N number of chromosomes are moved to the opposite pole, which indicates the reduction in the number of chromosomes. Number of chromosomes to half. That is, if you are asked with the question, what is the exact stage where there is a reduction in the number of chromosomes? Of course, it is meiosis 1. But in specific, if they ask, then your answer will be, it is Anaphase 1, at which the number of chromosomes are reduced to half. Here the half number of chromosomes and here the half number of chromosomes. If you observe here, both of them are present here together, but they are getting separated here. By the end of anaphase, they have reached to the opposite poles, and it is the beginning of telophase 1. So the next phase will be telophase 1, where in telophase 1 what happens? The chromosomes have moved to their opposite pole completely, and the chromosomes move to opposite poles completely, and formation of nuclear membrane, appearance of nucleolus, and the cell organelles associated with the nuclear membrane also appear, and there is an uncoiling of chromosomes. But here, exactly, the chromosomes are not completely uncoiled if you imagine it with the interphase. Like that, the chromosomes will not uncoil, just they uncoil, but they have their integrity. They will be straight like this. They will be having their integrity, but the complete uncoiling of chromosomes will not take place in the telophase. The chromosomes remain straight. The complete disintegration or complete uncoiling of chromosomes will not take place in telophase 1. And after telophase will be the cytokinesis, where there is a division of cytoplasm. The cytoplasm will divide. In case of animal cell, it is formed by the formation of a furrow, and in case of animal cells, the division will take place. The division of cytoplasm takes place by cell plate formation. So this, as a result, there is formation of two daughter cells having n number of chromosomes. By the end of this cytokinesis, there will be formation of two daughter cells which are having n number of chromosomes. Initially here 4 chromosomes are present in one cell, as we taken But here, by the end of cytokinesis, here is the cytokinesis happened. Imagine already. This cell has got 2 chromosomes. This cell has got 2 chromosomes. The number of chromosomes are reduced to half. So, the reduction in number of chromosomes will take place in the meiosis 1. After cytokinesis, the cell will spend some time in rest, and that resting phase is called interkinesis. We are referring here interkinesis because there is no replication of DNA taking place. DNA replication will not take place, but cell will rest for some time and immediately it will enter into the second step of division, that is the meiosis